Hello again. In the previous video, we talked about the issue of determining the number of genes in the human genome. In the Fields paper in 1994, suggested several ways to address this issue. If you look at my notes, if you sequence the genome, this is the point of me showing this figure. Now just imagine that you just sequence a genome and you've got the data, and what you see are these sequences. Now, so where are the genes? Is there any gene in there? So it's not an easy task if you don't know what you're looking for. So in my notes, this is from a slide when I attended a course in BGI. So in predicting, in determining whether a particular sequence of a particular DNA sequence is a gene or not, you can do several things. One is, of course, you use ESTs. Remember what I mentioned about ESTs? ESTs are express sequence tags. They come from cDNA. So if there's a database, if you have a particular sequence and you compare them against the database like BLAST, then it'll say it's an indication that what you have is a gene. SAGE serial analysis of genome expression, tiling array. These are some specific methods that again they deal with mRNA and tries to predict based on sequence. You can do transcriptome analysis. Transcriptome basically just says that you are looking at all the transcripts. Now you can also do homology based. This is later when we look at the Human Genome Project, one of the goals, one of the strategies of the Human Genome Project is also to sequence other genomes. Why? Because they will be useful for comparative analysis. So whatever you get, I think we also mentioned in class about the term conserved sequences. So whenever you get a sequence, if it is conserved in different various organisms, that gives an indication that it's actually an important sequence and may constitute a gene. So you can do homology-based comparison of proteins of lattice species. There are various databases available: the genome database, the protein database, the mRNA database, so you can compare. If you can find a match that gives an indication that perhaps your sequence is conserved. Other than that, when all because at that time, when people say the information in genome databases are quite limited, they have to rely on prediction, and various algorithms and calculations and extrapolations were made, and they came up with different software applications, for example, FGNAS, GenScan, Augustus. Basically, they predict the sequence, whether it is a gene or not, based on several characteristics. For example, if this is high GC content, because most coding regions are high in A's and T's. If you can find CPG islands, that's another one. If what else? If you can translate without any interruption over a long sequence of DNA, this is the open reading frame. So based on all these various characteristics, that they will be able to predict with a certain amount of certainty. Then there is a software who looks the results of different various other softwares. For example, the Glean software, what it does is it combines. Here's an example from EST database. So that is the one in red, and it suggests that the red small boxes here are genes. They compare it with different organisms. This is in the plant genome, the grape, poplar, papaya, rice, and Arabidopsis, and they say that there are some similarities. Look, for example, certain regions of the ESTs they do not have a match. Okay, that region, for example, they do not have any predicting matches, or sorry, homology matches against what is known in the plant genome. And then these are the results from prediction from using the various different algorithms. Again, look here under the Augustus software, you will find that there is a gene that's predicted here, but is not is not found in whether by homology, is not found in EST, is also not found by other prediction. So from all that, they devise yet another software that looks through all that and only accepts the prediction if it is supported by other methods, for example. So because you can see this box is almost present in all other methods, so that's actually there. These small boxes, they don't appear, even this one appears twice, this one appears okay, this one appears like 4 times, so it is accepted. But if it just appears once or twice, it is not regarded. It's regarded as something that is not real. Okay, so that's a little bit about how to predict a gene.